Can you hear me?
Yes.
Okay.
Hi, Professor. Yes.
When nobody says anything, I'm never sure. Wait another minute or so. We've only got about half the class so far.
The quiz is today, right?
Yes, that's the first point I want to make is quiz 4 is today.
Okay.
It's the one I told you what the question is. There's just one question on it. Some people have taken it already.
I tried. It wouldn't let me in.
Should let you in.
Wait, the quiz is only one question?
Uh-huh.
What?
Can you tell us what the question is?
How would you?
Yeah, the question is transcription and translation. So I give you two sequences, two DNA sequences, one wild type and one mutant. And I ask you to transcribe them both to RNA. And then I ask you to translate them both to peptides. And then I ask you to tell me what type of mutation is it?
That's it?
That's it.
That was like a question on the test, I think. 3 points, 3 points.
This is this is 10 points. See for
Don't forget Yolanda Dukes.
uh-huh. Yeah, Yolanda, what's on your mind?
Oh, I asked you earlier when we first got on.
Okay, so that was your question. All right, so that's quiz 4. That's today. Quiz 5 is Monday. So quiz 4 was just one question, and it's asking you to do just one thing, two things actually, transcribe and translate. And quiz 5 is all the other things having to do with mutations and CFTR. So that will be Monday, and again, it'll be 15 minutes. There are only, I think, 4 questions on that quiz. And so you should look at the slides and also look at, remember you submitted questions and we discussed them last time.
All the quizzes are worth 10 points.
Pardon?
No matter how many questions, they're all worth 10 points.
Yeah, the quiz is worth 10 points, even though there are only 4 questions.
Okay.
And
Okay.
the quiz 4 is just one question, but it is still 10 points.
Okay. Okay.
So to prepare for the quiz on Monday, review the slide set on mutations and CFTR. And I took a couple of the questions from the discussion we had last time based on the questions that you submitted. So take a look at that. The questions and their answers are in module 8, I think, either 8 or 7. All right, we've only got 60 minutes today because you're doing a quiz. So I want to get going on that. And share my screen with you. And you should see CRISPR. Anybody not see that?
I could see it.
Okay, good.
Yes, we can see it.
So CRISPR is revolutionizing biology. All different aspects of biology. In fact, last year, the 2020 Nobel Prize in Chemistry was awarded to two scientists, one Jennifer Doudna at UC Berkeley, and the other Emmanuel Carpentier from France. They got the Nobel Prize for discovering and developing CRISPR. So that alone tells you it is a big deal. So we often think of mutations as things we don't want. So for example, CFTR, we don't want mutations in that gene because if they're the right mutations, they'll make us sick. But sometimes you want mutations. And so if you do, how do you get them? Well, as we've talked about before, mutations can occur as a result of errors in the copying of DNA. Things like radiation or various chemicals can damage DNA. We talked about thymine dimers formed by UV light. And then there is CRISPR, which is what I want you to have an appreciation for. And that's why I'm spending a little bit of time introducing you to it. So what is CRISPR? So if you want mutations, you could wait for them to occur naturally, but you might wait your entire life and never get what you want. Or you can use mutagens like UV light or chemicals, and they'll make mutations. They'll make a lot of mutations, but they're random all across the genome. And so once you've made your mutations, then you have to spend a lot of time and effort sorting through them to find the one you want. And that's what makes CRISPR so great. So I have here in the box why CRISPR is so important. So this is what I want you to remember. CRISPR permits any mutation to be made in any organism. So in simple words, it's first of all site specific, and secondly, it's universal. And that will become clear as we go on to the next slide.
Professor, real quick,
Yeah.
what do you mean specifically by universal? This is Nicholas Gonzalez.
Okay, yeah, that'll become clear in a bit, I think, but let me just elaborate now that you've asked. Site-specific means you can target any gene. It doesn't even have to be a gene. You can target any sequence, an entire genome, and say, that's the sequence I want to make a mutation in. Universal means it works in any organism. And this is truly astonishing, that whether you're working with a bacterium, a mouse, a plant, a fungus, an animal, a whale, doesn't matter what organism you use that you're interested in causing mutations in, CRISPR works.
Professor, this is a little off topic, but this is Alex Salos. I had a quick question regarding the worksheet on the cystic fibrosis pancreas. What are we doing? What is it? What are we supposed to do?
We'll cover that today. I hope we get to it today. Yeah, don't worry about the worksheet until after we've gone over the materials. You've got, I think, 5 documents in module 8. And those 5 documents, if you read them, give you a lot of information, including the answer to your question. But I'll go over that later in this presentation.
Okay.
Okay. Elizabeth, did you have a question?
Yeah, it's about CRISPR. That's how you pronounce it, right?
Mhm.
You said you can target like anything, right? Technically, like
Yes.
any. So I think I heard, I don't know if I'm wrong, but I think I heard this like a while back from CRISPR, that you could, like, people were saying that at one point in the future, we would be able to actually, like if a baby was gonna be born, like change their eye color and stuff like that.
Uh-huh.
Is that what this is?
Yes.
Oh, okay.
And now you're actually going beyond the scope of this course and talking about ethics.
Oh, yeah. I think
Because
I had a conversation in class with my old. I was in medical, in a medical pathway, like science pathway, and we
Uh-huh.
talked about that aspect of it. But I didn't know if that was this. So
Yeah.
yeah.
Well, if you want to see a full-fledged discussion of that, Jennifer Doudna has written a book. Can't remember the name of it, but if you look up her name, Google her name, you'll see her book. And she has at the end of it quite a long discussion about the ethics of using CRISPR because it is so powerful and because it is universal. So right now, the use of CRISPR on humans is not allowed. It's been
Makes
done
sense.
one time in China. The Chinese scientists did it surreptitiously, and he is now in jail. So it's really a no-no to do it in humans.
You can see why.
Probably for the best too.
Yeah, because
Best,
we
I
can
agree.
basically, we have the power now to completely change the nature of our species. And of course, we can change the nature of any other species, which we care much less about than our own. Okay, let's go on. I want to tell you what it is and why it works. So this picture on the right is what I want you to remember. On the left are definitions. What is CRISPR? It's a cluster of repeats, DNA repeated sequences that occur in bacteria. And the purpose of them is to remember the sequences of viral pathogens. So if a virus invades a bacterial cell, the bacterial cell will eventually recover from it, but it wants to remember that. And so it uses this cluster of repeated sequence as a memory mechanism. Now, the other part of CRISPR is an enzyme called Cas9. So to edit a genome, to do all this powerful stuff, you need just two things. A piece of RNA called the guide, and you need the enzyme Cas9. So how does it work? On the right, first you make the guide, a piece of RNA, and you can synthesize this in the lab. You can make it, it can be fairly short, 10 or 15 nucleotides, or it could be considerably longer. But once you've synthesized this piece of RNA, you mix it with the enzyme, Cas9. And then you introduce that into the cells whose genome you want to mutate. So what happens is this guide RNA finds its target in the DNA of the cell. And when it does that, Cas9, if there's this little yellow sequence on the end, Cas9 will make a double-strand cut, which you see here, and that is a mutation. Once that cut has been made, you have a number of alternatives. You can leave a gap there. In other words, that would be a deletion. You can insert a different piece of DNA there. That would be an insertion. You can introduce a frame shift. Almost any kind of mutation that you would like, you can do. And you've done it at this site that you've targeted. You start out saying, what in the genome would I like to mutate? Then you make a guide RNA that corresponds to that. And that's what makes it specific. The guide RNA goes only to the sequence that you want targeted.
Question, Professor,
Mhm,
as Steve Stenson.
mhm.
So if you can do that, then why can't they do something about Lou Gehrig's disease? Because it's illegal?
Well, on humans, yeah, it would be. Although people are starting to do that, what's forbidden is to make mutations in the germline. But if you want to correct a mutation in the autosomes in the body cells, not in the reproductive cells, for therapeutic purposes, people are working on that right now. And in fact, I'll talk a little bit about that with respect to CFTR. If you could correct the mutation in CFTR, then in principle, you would cure the disease. But we can talk about that a bit later. I want to make sure you understand how CRISPR works, that you need these two things, the guide RNA, Cas9, the enzyme that makes a double strand cut. This is just an elaboration of the components of the CRISPR system. And you can see here at the bottom of this center panel that you can make an insertion, a deletion, a frame shift, or actually other kinds of mutations once you've targeted the site. So to reiterate, CRISPR has this extreme target specificity, and it works in any organism, bacteria, archaea, viruses, eukaryotes. It seems to work wherever you try it. People are even talking about targeting the coronavirus to see if it can be used as a therapy for coronavirus. And as I just said, it might be able to correct genetic flaws such as those that cause cystic fibrosis. And why should you be knowledgeable about it? Well, one of you has already mentioned you've discussed it, you read about it in newspapers and online almost every day. There's something about a new use for CRISPR. And it's very likely that it will affect you or somebody you know sometime in the future. So you should be aware of what CRISPR does and how it works. And on that note,
Professor, I have a question. This is Gabriela Galvez.
okay.
Professor, the CRISPR helps with the multiple sclerosis, could be? Because I see the muscular dystrophy, the CRISPR help the muscular dystrophy, right?
Uh-huh.
And what about the multiple sclerosis? So sickness like that, could the CRISPR work on them?
In principle, any disease that's caused by a mutation, in other words, any heritable disease, in principle, could be treated with CRISPR. There are the ethical considerations, and as of right now, you can treat body cells, but you can't treat germ cells with CRISPR.
Have there been any
Thank you.
clinical trials with the CRISPR being used by any chance?
I'm not aware of any. I know that some labs are working experimentally, trying to use CRISPR, I think even on CFTR, but I'm not aware that there are any clinical trials. There might be, but they're probably proprietary and nobody will know about them until the results are in, and then only if the results are good.
All right, thank you. This was Nicholas.
Gonzalez, okay. All right.
Is CRISPR the thing that I hear about in the news where they are trying to bring back extinct animals, or is that something that's different?
Yeah, that's different. To bring back like the woolly mammoth is in the news.
Yeah,
these
that's what I'm
days.
talking about.
And that's, yeah, that doesn't involve CRISPR, at least not directly. What's involved is, first of all, you have to get the complete sequence of the woolly mammoth. So you have to isolate fossilized DNA and sequence it. And once you have the sequence, you would try to put the complete sequence in a cell and get that cell to divide and undergo development, and hopefully a mammoth would emerge. Now, this has already been done in bacteria. You take a bacterial cell and take out its DNA and replace it with artificial DNA that you've created in the lab, and the cell actually works. The new cell that you created actually works. So we know that it's possible to do it with a single cell. What will it take to do a multicellular animal like a woolly mammoth? You know, that's a whole different matter, and it's a big if, but
Sounds complicated.
it would be very complicated and it would take a long time. And who knows, because technology advances at such a rapid rate, it probably will be possible at some time to, I don't know about woolly mammoths, but to create extinct animals from.
So is that still like what we talked about earlier in the session? We were talking about animals, or that movie where they put those different segments in a dog that made him do other animal characteristics? Is that the same?
That's not the use of CRISPR, but it's the same thing as trying to bring a woolly mammoth back.
You're taking different
Professor,
DNA from other animals and you put it into one to make him be a special type of animal or mechanism that could be.
Yeah,
or
and this is done with experimental animals like fruit flies, for example, are working models. So you can make a fruit fly that has eyes instead of on its head, it has eyes on its abdomen that are perfectly functional eyes just by changing regulatory genes.
Is that same thing like
Professor,
where they have those those lions that are huge? They're just huge. I mean, how do you get their regular size to be 10 times the size that they really normally are? Is that part of splicing?
Yeah, that's a cross between a lion and a tiger.
To be that big?
Very often when you make a cross between a lion and a tiger, you'll get an animal that's called a liger that is actually bigger than either of the parents.
And that's real?
Yeah, that actually has been done.
Well, don't forget my name too. So that's Yolanda.
Okay, I got it.
Professor, this is Gabriela Galvez again. Sorry, it's because I just want to know if the CRISPR is used for the cancer research, talking about the cells.
Yeah,
Yeah,
I kind of had a similar question with that because it's like specifically targeting, no?
yeah, and
Yeah.
cancer is a genetic disease. So any genetic disease, in principle, could be treated with CRISPR. Cancer is a much more complicated genetic disease than cystic fibrosis. Cystic fibrosis is a single mutation. In cancer, you have to have multiple mutations in order for a cell to turn malignant. And so to treat that, where you'd have to deal with a dozen or more mutations and correct them all, that would be very complicated. But the technology is there to do it, or at least try to do it.
This is Aiden Heinsley.
Well, how is it that you have cancer and they do surgery and they take away, as they say, some parts of it that is gone, and then it comes back? How is that possible?
Well, if it comes back, it probably means that the original cancer metastasized. So if you have cancer of a particular organ, say the liver, and you can do a liver replacement, get rid of the cancerous liver and replace it with a healthy one. But if the liver cells, the cancerous liver cells have escaped and now they're elsewhere in the body and continue to divide, you still have cancer.
So then, Professor, could it also be said that it's not just cancer itself, because there are other kinds of cancers and also there's different stages of cancer? So it's not as simple as saying, well, let's use this just for cancer. It's like, well, it's not as simple as that, more complicated.
Yeah, cancer is very complicated because even if the malignancy remains confined to a single organ, it's constantly changing genetically. So what people can do now is sample a tumor and sequence the DNA of the cells in that tumor. Now, if they come back a week later and take another sample and sequence that, they find that it's different. So cancer cells mutate and mutate and mutate.
But that makes sense out that they, with like CRISPR has these different types of experiments he's using or trying. And out of all the years that they've been trying to find an actual cure for it, and yet there is no cure at all for no cancers.
Well, I wouldn't say that. Some cancers can be successfully treated. Some people have been in remission from their cancer for many years. For example, prostate cancer in men. A lot of men have had prostate cancer, have had their cancer treated, and they've been cancer-free for decades.
But when you say you get rid of it, means that you won't, it's gone for good. That's what I said with all the experiments and the all the research that they're doing and have been doing since before my time or way before we could think of, and yet there's still no set cure. You can just say, okay, we can go in the hospital and you don't have it no more at all. It's over. Like with John Wayne, he was cured for so long, but yet he still ended up dying from it.
It depends on the person, the cancer, the particular circumstance. In some cases, cancer is successfully treated. In other cases, it's not. Sometimes it looks like it's been successful, but then years later, the cancer comes back. So there's no universal cure for cancer, but much progress has been made against certain cancers. And prostate cancer is a good example of that. Certain breast cancers are other examples where women have been treated for their particular mutation and have been cancer-free for a long time.
Professor, I have another question. Like you say, this is the CRISPR came to revolutionary biology. It is something wrong on CRISPR or no?
Say that again.
It is something wrong with CRISPR? Something wrong?
Is there something wrong with it?
No. Can we find something wrong with CRISPR?
Well, if you're asking an ethical question, can CRISPR be misused? The answer is yes, it could be misused. And scientists are very much aware of that. Ethicists and philosophers are very much aware of that. And it's a topic that's being debated, probably as we speak.
But by talking about the DNA, nothing can be wrong with the CRISPR?
Oh, can CRISPR make a mistake? And be non-specific? It's possible. Yeah. And so great care has to be taken when somebody makes what they think is a site-specific mutation. They have to do follow-up work to make sure that they have not caused any mutations anywhere else.
Thank you.
This is Aiden Hinesley. I was gonna ask, is CRISPR the name of a process or is it something in a cell?
It's a series of DNA repeats that were originally found in bacterial cells. And you see here on this slide what CRISPR means. It's clustered, regularly interspaced, short palindromic repeats. That's what CRISPR stands for. And Cas9 is CRISPR-associated protein 9.
Yeah, so how did that play into like the picture or model of how CRISPR works?
Well, that's what the Nobel Prize was awarded for. So when people first noticed these clustered palindromic repeats, and we'll talk about palindromes later, what they are, they didn't know what to make of them. They were just weird. And so it took a lot of study, especially by the people that won the Nobel Prize, to figure out what they were doing, that they were really designed as an immune system against viruses, and then to take components of the CRISPR system from bacteria and develop it into a simple system like you see here, where you just need two components. You need this one enzyme and a synthetic RNA, and that's all it takes to make the mutation that you want. Okay. I want to.
Will we need to know that what you said CRISPR mean and the Cas9 for the quiz for next week?
No,
Okay.
I don't think that's CRISPR. I don't think I put CRISPR on the quiz. If I did, it's a very simple question about it.
Okay.
Now I want to switch to cystic fibrosis. You'll find this document, hope you can see it, Cystic Fibrosis Introduction. You'll find this in Module 8 in Canvas. There's also a 2-page document there from the National Library of Medicine, which is a very nice summary of this disease. So, and I've got 3 points here taken from that summary. First of all, cystic fibrosis is a genetic disease. It's inherited from both parents. The main symptoms are in respiration, that is breathing, and in digestion of food. It's fatal, usually in childhood, if it's not treated. There are now, and you'll find this when you research your project, that there are quite a number of treatments for cystic fibrosis, which allow people with it, who are born with it, to develop into adults. As adults, men are infertile and women have difficult pregnancies. So people who have cystic fibrosis are unlikely to pass it on to their children because they can't have children. Now, cystic fibrosis can affect almost any organ in the body, but the main ones are the lungs and the pancreas. In the lungs, as you know, lungs have these little air ducts. If they fill up with mucus, it interferes with the absorption of oxygen, and breathing becomes very difficult. In the case of the pancreas and the gallbladder, there are these little ducts we'll talk more about in a minute, these ducts get clogged up with slimy, sticky stuff. So the digestive juices cannot pass into the intestine. Now, someone asked, we talked about this last time. One of the questions that was submitted is what actually happens in cystic fibrosis. And here, item number 2 compares a healthy individual with one with cystic fibrosis. Now, in the case of a healthy one, you have, this is an organ, say a pancreas. These cells are on the epithelium on the outside of the pancreas. And on the inside, you have water and chloride ions. And in a healthy cell, the CFTR protein transports chloride ions across the cellular membranes to the outside. And when that happens, water goes with it through another pathway, but water accompanies the chloride ions, which keeps this mucus layer hydrated, keeps it fluid, so that these cilia on the cell surface can sweep away things like bacteria that you don't want. Now,
I have a quick question.
Yeah.
Is the bluish background, that is the mucus layer in this, right?
Yeah, the pale blue is a diluted mucus layer, and on the right, the darker blue is a thick, sticky mucus layer that's deficient in water.
Okay, but the mucus layer is always there. It's just not as thick, essentially, like gelatinous.
Yeah, yeah.
Okay, gotcha.
Mucus is everywhere in our body.
Yeah, yeah.
And people with cystic fibrosis have, instead of having healthy mucus, they have thick, sticky mucus, which makes their lives very difficult.
Gotcha.
And the reason for that is that without a CFTR protein, or with a defective one, chloride ions are not transported, water is not transported, and this mucus layer gets very thick and sticky. The bacteria cannot be swept away. They form a biofilm, which can actually be infectious. So that's what causes the symptoms, regardless of the organ that you're talking about, whether it's lungs or pancreas or testicles. That's what causes the symptoms, this thick mucus. And the third point here I want to make is about the inheritance. How does one get this disease? Well, both parents have to have one mutation. So the father has to have a healthy chromosome 7. Remember, the gene is on chromosome 7. And one mutated CFTR gene on chromosome 7. The mother has to be the same, one healthy and one mutant. So neither of them has any symptoms. They don't know that they are carrying these mutations, but they can pass them on to their children. So one thing that can happen is that the father contributes a healthy chromosome, the mother contributes a healthy chromosome, and this is a normal, healthy child. Or the father contributes a healthy, the mother a mutant. That's a carrier, it's carrying a mutation, but it's still healthy. And the opposite can occur. The father can contribute a mutant and the mother a healthy. That child is healthy, but a carrier. If a child gets both, a mutated chromosome from the father, a mutated chromosome from the mother, that child has cystic fibrosis. And you'll deal with it. You'll each get a mutant to study, and in all cases, this is the inheritance of the mutant you'll be looking at.
Excuse me again, Professor. I hate to ask this, but is this similar to how sickle cell anemia is transmitted between parents?
Yeah,
Okay.
I'm not sure if it's exactly the same, but yeah, you have to be your parents, one or both, have to have the disease in order for a child to express it. Okay, let's go on to the pancreas and the gallbladder. The pancreas is this yellow object here, and you can see that running down to the middle of it is a duct called the pancreatic duct. The gallbladder, I've drawn a little picture of it here, and here this green tube is bile duct. They both empty into an organ called the duodenum, which is the beginning of the small intestine. Now, if you look inside the pancreas, there are two types of cell. There are exocrine cells and endocrine cells, and you'll need to know that. Exocrine cells release substances out through the duct. Endocrine cells release hormones into the bloodstream. So the substances from exocrine cells go into the intestine. Endocrine cells release hormones into the bloodstream. So this picture here shows the pink cells with the blue dots. Those are exocrine cells, and they produce pancreatic juice. And we'll talk about what's in the pancreatic juice in a minute. The blue cells are called the islets of Langerhans, and they are the endocrine cells which produce hormones. So you have these two functions in the pancreas. Some cells produce digestive enzymes, some cells produce hormones.
Real quick,
Uh-huh.
someone in the chat asked, it was Alexandria Parrish, she said, or he, is it possible to get a genetic disease and never know about it because the person never displays any of the symptoms?
If you're a carrier, yeah.
Mm.
You don't have the disease if you're a carrier. So if you go back here to these chromosomes, these children that get one copy of the mutation, but the other copy is healthy, they never know that they're carriers. Because they have no symptoms. Only if a child gets two mutant copies, symptoms develop. That's the only way they know that the parents both were carriers. So let's first look at these ACE&R cells, these pink ones. They're the exocrine, and they produce digestive enzymes. What are these enzymes? Well, think about the food you eat. What's it composed of? It's composed of proteins, fats, sugars, and some nucleic acids. So pancreatic juice has enzymes for all of those. Proteases for proteins, lipases for fats, carbohydrases for sugars, and nucleases for nucleic acids. These enzymes are all produced in the exocrine cells, these ACE&R cells. However, scattered throughout the pancreas, so throughout this mass of cells in the pancreas, are the endocrine cells that produce hormones. What are the hormones? Well, as you might suspect, one of them is insulin. So about 70% of the endocrine cells produce insulin. And what that does is promote glucose absorption into the liver from the blood. Another hormone called glucagon does the opposite. It raises blood glucose level. So between insulin and glucagon, they regulate the level of glucose in the blood. Another hormone, somatostatin, produces a very minor amount of hormone, but it regulates the effects of insulin and glucagon. And then there's another one called the pancreatic polypeptide, which again has a regulatory function. It's a very small component of all the cells in the pancreas. So the pancreas produces over 1 liter of juice each day to help you digest your food. Liter is about 1 quart. And this juice contains all these enzymes that we've just talked about. And it's also buffered against the acidic gastric juice that comes from the stomach. And I'll talk about that in a minute. And this brings us, in red here, is the word zymogens. That's a word you'll have to know. I'll ask you about that at some point. Zymogens are inactive forms of enzymes. So the pancreas produces proteases. These are the protein digesting enzymes as zymogens. So they're produced as inactive zymogens in the pancreas, but when they get to the duodenum, they're activated. So now they can start digesting protein in the food. If the proteases were produced in the pancreas in an active form, they would digest the pancreas, which would not be good. And that's why they're produced as zymogens, as inactive enzymes. And they don't get activated until they actually leave the pancreas. But here are some examples of zymogens and their corresponding peptidases. Trypsinogen is activated to trypsin. Procarboxypeptidase to carboxypeptidase, and chymotrypsinogen to chymotrypsin. So these activated enzymes occur only in the intestine, not in the pancreas. So let's take a look at the larger picture, the whole system, how it works. So we're going to look now at 4 different organs: the stomach, gallbladder in green, pancreas in yellow, and duodenum in pink. But when you eat food, it goes down your esophagus into your stomach. And there, it's met with a very harsh environment, which is pH 2. pH 2 is extremely acid. It's only slightly less acid than hydrochloric acid, pure hydrochloric acid. So something for you to think about, we can talk about it next time, is why is the stomach pH 2, such a low pH? And there's this enzyme in the stomach called pepsin. It's a protease which has a pH optimum at 2. So after the food is partially digested in the stomach, it empties into the duodenum. Here's where the real digestion starts. So the pancreas is producing these 4 different types of enzymes, and the gallbladder is producing bile. Well, you know what the enzymes do. They digest the food, but what is the function of bile? Well, if you go to the panel on the right, the gallbladder produces bile, a complex fluid that contains water, electrolytes, and organic molecules, including bile acids. What are bile acids? Well, an example here, cholic acid. So you can see the structure of it here. When you see the yellow part, what does that immediately tell you it is? It's a steroid. And steroids, as you should recall, are highly non-polar. They're very hydrophobic. However, that's cholic acid, but there is another molecule called glycocholic acid, which is modified by what's circled here in red. The amino acid glycine is added to one end of it. So that means that one end of this molecule is highly hydrophobic, and the other end is hydrophilic, water-loving. That is the definition of an amphipathic molecule. Amphipathic is a molecule that's hydrophobic on one end and hydrophilic on the other. Well, why is this important? It's because a good deal of our diet is fat. And fat is not soluble in water. And since digestion occurs in an aqueous environment, we couldn't digest fat, the fat that we eat, if it wasn't solubilized. That's what bile acids do. Glycocholic acid, this amphipathic molecule, it has a non-polar end that goes in the fat, has a polar end that goes into the aqueous phase, and that causes emulsification of the lipids so that they're now soluble in water and they can be acted on by lipases and broken down into molecules that we can use as energy. So the big picture here is that these two organs, the gallbladder and the pancreas, collaborate. The pancreas produces enzymes that digest food, and that's all fine except for the fats. The fats cannot be digested until they're solubilized, and that's what the gallbladder does. Produces bile acids which solubilize the fats that we eat. Any questions about how the system works?
Well, yeah. Ooh. Hello. I
Yeah.
was asking about the gallbladder. Wait a minute, where do we go to that screen? Oh, here we go. The gallbladder, you said it functions with the pancreas, but what is it? Sometimes they say they take away the gallbladder completely. So
Then you
how
have
does
to
that
watch
work?
your diet.
Huh?
So either you supplement your diet with enzymes that replace the glycocholic acid or the bile acids, or you try to keep fat out of your diet. You try to derive all your energy from other substances. One thing I neglected to say is that it's for zymogens, it's only the proteases that are produced by the pancreas as zymogens. The other enzymes don't act on proteins, so they can be produced in the pancreas as active enzymes. It's only the proteases that are zymogens. Okay, this document is in module 8, and you can refer to it, study it, along with several other documents that will help you. I'd like to stop sharing this and go to the next document.
Excuse me. So
Yeah.
this document that you show us gonna be on the quiz 5.
No, it will not be.
Oh, okay.
No, this moves into the next topic. Okay. you down. All right, I'm going to share again. This is your project. And again, you'll find this document in module 8. So the first part of this is a whole series of websites which give you background about cystic fibrosis. So you should check those. A really good one is this one from the Cystic Fibrosis Foundation, which is shown here. And now I'd like to, in the time, I don't have much time left, but briefly introduce you to what your project will be. And I should point out here that this website is just a starting point. You'll really need to do searches, for example, Google searches or searches of the National Library of Medicine in order to find all the information you need for completion of your project. Now, here are a list of mutants. You're going to be divided into groups, and each group will be assigned a mutant. This project is worth up to 100 points per student. And I've made a circle here around this point that each student must advise me by Monday, October 11th, that they want to participate. You may wonder, why am I asking that? And the reason is, I found from past experience that if I just assign everybody on the roster to groups, some people don't participate. And that's a huge frustration for the members of their group that are participating. So I want to assign people to groups only if they've indicated that they want to be involved. So please let me know before Monday. Send me an email and let me know that you want to participate. And anybody that doesn't, by October 11th, I will not assign to a group. So the class will be divided into these groups, each group assigned a mutant. And then each group will make an oral presentation to the class, which is illustrated by no more than 10 PowerPoint slides. The timing of the presentation, I don't know how many groups yet, and so I don't know what the timing will be. I'll let you know as soon as I make groups, but it must be precise, and I'll deduct points if you either go way over or way under your assigned time. Now, the other requirement here is that you have to have a preview of your presentation at least a week before delivery to the class. And once I know how many groups and who is in each group, I'll set up time slots so that you can reserve a time to preview your presentation with me before you present it to the class.
I have a question, Professor.
Yeah.
What if we don't tell you that we don't want to, like, what if we say we don't want to participate in this? Like, is there an alternative assignment, or?
No.
You just get a 0?
You miss the possibility of 100 points.
So we get a 0.
Yeah, you simply don't get points that people who do participate will get.
So all the groups that will participate, they will be at the same day?
I think so. It depends on how many groups we have. But I think, yes, we'll have a day for presentation. So each group will will present their slides and their oral part by Zoom to the rest of the class.
So we just let you know by email, right?
Yeah.
Okay, perfect. And Professor,
Professor?
bring us into the group.
So when is the deadline of the project?
I don't know yet. I'll let you know as soon as I find out how many people are going to participate.
Okay, so are you gonna assign the group or we have to choose the partner?
No, I'll assign people to groups.
Oh, okay.
This will be a random process. So that, I'm not predicting, or I'm not doing it by alphabetical order or any other order. I'm just randomly assigning people to groups. And I'm not even sure how many people will be in each group, probably either 3 or 4.
Okay, thank you.
And so here's your assignment then for your group. What is the effect of your mutation on the structure of the CFTR protein? What is the effect of your mutation on the function of the CFTR protein? What are the symptoms that your mutation causes? And what are possible cures or treatments for the symptoms caused by your mutation? And then I will grade you on adherence to following instructions and on the quality of your slides. And I've got a website here that gives you some pointers about how to make slides, make effective ones. And so then the quality of your oral presentation and a sound conclusion at the end. Now, I've bolded here why I think this is an important experience for you, because it will help you to gain experience in researching a topic. You will gain experience in how to prepare illustrations for your topic and how to deliver it to a group. So in the past, people who have been involved in this have felt like they benefited substantially from the various elements in the project. And finally, I have some of my own tips for making slides here at the bottom of this page, which I won't go through now because I'm out of time. And so we'll have to quit for today. And remember, if you haven't taken the quiz already, do it. And there'll be another quiz on Monday.
Wait, there's gonna be a quiz 5 on Monday?
Quiz 5 is on Monday. Yeah, it covers the things that we haven't covered yet. The quiz you're taking today has just one question, asking you to translate and transcribe. Other aspects of the questions that we discussed last time and the slide set on mutations, they're covered by the quiz that will be on Monday. As I said, there are only 4 questions in that quiz.
Okay.
Okay, anything else?
Professor, I have a question about the today quiz.
Uh-huh.
So the quiz, I mean, I just saw that there's only one question. So it's going to be written question or just multiple choice?
Pretty good.
There's only one question today, but it has several parts. So I'll give you 2 sequences, 2 DNA sequences. One is wild type and one is mutant. And what I ask you to do is to transcribe each of those DNA sequences to RNA, and then translate each of the RNA sequences to a peptide.
Okay.
So you'll have to, and so you'll have a codon usage table in the quiz that you can use.
Is there like anything for today's quiz like we could look at to like help us? Like, is there any slides or anything like that?
Yeah, the last lecture had several slides about it.
Okay.
And we've talked about it several times already how you do translation and transcription. And that's really all I'm asking you to do here, is to go to do with a new sequence what you've done already several times with sequences from class.
Okay, so
And the last part of the question is, what type of mutation occurred?
okay, so just the last question, just to be sure, you said everything module 8 will help for the homework assignment this week, right?
Yeah, the assignment about cystic fibrosis, you should be able to answer all those questions based on what you heard today and on, well, on that. There are several other documents in Canvas that you should read, and they will all contribute to your understanding of how to do the worksheet.
The assignment doesn't seem to have questions. It sort of just seems like you admit.
The worksheet has 12 questions. If your worksheet doesn't have 12 questions, there's something wrong, let me know. Okay, anything else? Stop sharing.